Okay. So, started talking about proteins last time. And it looks like we just barely got started. We created a definition. And the thing that I'm most concerned about or most wanting you to be aware of, like with the other groups of macromolecules, I need you to be aware of the functions that these perform. And so we have 8 functions that we're concerned about. That's a lot of functions. Proteins do a lot of things. And when we start to look at the structure of the proteins, we'll figure out why it is that proteins can do all of these amazing things. So we have structural proteins. Again, keratin and collagen are examples of those. Biological catalysts, these are enzymes. We've talked some about those. Transport proteins, both in the blood and in cell membranes. So that's 2 categories right there. So 1, 2, 3, 4. And then cell communication you're already familiar with from carbohydrates. Chemical messengers, these are hormones and neurotransmitters. And then cell membrane receptors and immune proteins. And we want to be able to figure out why these things can do the things that they do. All of our other macromolecules aren't really that impressive when you start looking at proteins. Proteins are far more complex. They can do far more greater numbers of things than what our other molecules do. So we know that we have 2 energy molecules, right? We have glucose and we have our carbohydrates and our fats. We know that sugars also participate in the sugar phosphate backbone of ribonucleic acids and deoxyribonucleic acid. We know they participate in cell communication. But we also know that lipids can participate in the cell membranes and some types of chemical messengers. But we've got all of these functions here that are going to be explained by the structure of the proteins, whereas most of the other molecules that we've talked about are basically just carbon and hydrogen with a little bit of oxygen thrown in, and they're basically linear types of molecules. But in order to actually accomplish functions that enzymes accomplish, we need to have these amazingly complicated 3-dimensional structures. Most of the other things that we've talked about so far are just 2-dimensional or linear kinds of things. And I'll show you what I mean by that when we start to talk about the structure here. And we said that as far as proteins go, they're composed of monomers called amino acids. And we remember that we had, on one side of an amino acid, we had the acid, right? The COOH group is the acid part of this. And on the other side of this is the amino group, the nitrogen with 2 hydrogens on it. And I want to just sort of make a point here. In the carbohydrates, we talked about the idea. Carbohydrates, we talked about the idea that we got a little bit complex, okay? Here's our sugar molecule, but that we hooked our sugar molecules together through these OH groups, okay? Remember, we created dehydration reactions, and we hooked these together through these dehydration reactions where we took a water molecule out. With fats, we had a glycerol molecule, and we hooked them together with fatty acids through the fatty acid portion here, and we take out a water molecule and we hook the fatty acids to the glycerol molecule like that. And with nucleic acids, we had a different type of a linkage. The sugars, the 5-carbon sugars, were linked together through phosphate groups. Okay? So hopefully it makes sense that some of the function that these molecules have relates to how they're hooked together. We hook together OH groups to connect sugar molecules together, and these are basically storage products. We hook OH groups with COOH groups together in fats, and these are also storage products, energy storage products, or they're lipids that we use in cell membranes. With nucleic acids, we hook things together with phosphate groups, and we'll talk more about that when we talk about DNA and RNA and how genetics work. But when we hang on here. So when we look at amino acids, we see that they're going to hook together in yet another different way. So I've got the OH, or I've got the acid part of the amino acid right here. And the point that I want to make is that these guys are going to hook together by taking a water molecule out. But now I'm going to join this nitrogen, or this carbon that has the oxygen double bonded to it, to a nitrogen. And this is going to make all the difference in how these proteins are able to function. And I'll tell you just exactly what it is about this linkage here, this bond, that makes it so that these molecules can be these wildly complex three-dimensional structures, whereas everything else is basically just a two-dimensional molecule that doesn't have nearly as much function as these things do. Okay, so that's kind of the point I want to make. So start looking for that as we build these proteins. And I really apologize for my nose. And I'll continue to apologize for my nose. So keep looking for how this structure is going to reflect the function here. So remember, before what we said about the structure of amino acids, their name gives you the clues to remember the important features. Remember, we said because of the atomic structure of the carbon atom, we can picture this as having 4 valence electrons, right? And we can even draw a little diagram like this with 4 dots around it that shows us those 4 valence electrons. But for some carbon compounds, we can visualize a central carbon with 4 things around it at these 4 positions. And we had this structure, and this is what makes all the difference in the world for carbon. This is why carbon can do so many things that no other atom can, and that's why Sperry and anyone else who talks about biology and atoms in biology, we always start with carbon, right? Because carbon can do so many things. So for amino acids, again, the word amino tells us that an amino group, that's this right here, a nitrogen with 2 hydrogens, is one of the things bonded to the carbon. And we'll put that at the 3 o'clock position. The word acid tells us that an organic acid, or COOH group, is another thing bonded to the central carbon, and we'll put that at the 9 o'clock position. At the 12 o'clock position, we'll put a hydrogen. And all 3 of these things, the amino group, the organic acid group, and the hydrogen, are the same for every amino acid. Does anybody know how many? Oh, well, it's right there in the next statement. We have 20 different amino acids that can be added together to make proteins. This means the thing at the 6 o'clock position has to be one of 20 different things. Okay? Now in math, if we say that something can have different values, we call that a variable, right? And we do the same thing here in chemistry, except the variable is not going to represent a number, it's going to represent a chemical structure. And don't worry, you're not going to have to know these 20 different chemical structures. You just need to know that we have the name R group. We're going to put R there, and that means, hey, this can be one of 20 different things. Okay? Again, you're not going to have to know those. Now, having seen how carbohydrate monomers are linked together to form carbohydrate polymers, again through the OH groups, and then how fatty acids are linked together to glycerol to form fats, this suggests a way to us as to how amino acids get linked together to form a polymer of amino acids. And what we're going to remind ourselves is that mechanism is and I don't know why I have 3 lines here, a dehydration reaction. That's the mechanism of hooking amino acids together, a dehydration reaction. And remember, I've told you that dehydration reactions are one of the single most important ways that chemicals can interact in the cell. We have to do this again and again and again and again. And this requires a different enzyme for each amino acid. So if I want to hook this amino acid to this amino acid, I'm going to take one of the hydrogens here and the OH away from the acid, and I'm going to use an enzyme, and that enzyme is going to cut those out. Okay, it's going to cut those out, and that's going to leave me with a nitrogen that has this other hydrogen here hooked to the carbon that's double bonded. Okay, and we're going to call this a peptide bond. You don't need to know this designation, but this is a peptide bond. The nitrogen from one amino group hooks onto the carbon from its neighboring acid group. Okay? And I've already mentioned this. In carbohydrates, the linkage is between 2 OH groups. In fats, it's between an OH group and a COOH group. And in amino acids, it's between the COOH group and the NH2 group. And this is the difference between this and everything else that makes it be able to have 3-dimensional structures. So, in proteins, the main purpose of the amino acid group, or the amino group and the acid group, is to provide a way of linking amino acids together in long chains. And again, we've said this type of bond is called a peptide bond. And I will want you to know the word peptide. So in saccharides, we said if I had one saccharide, that was a monosaccharide. If I had 2 saccharides, that was a disaccharide. If I have 3 saccharides, that would be a trisaccharide. If I had more, it might be an oligosaccharide. If I had even more, it might be a polysaccharide. What do you think I'm going to call the linkage of 2 amino acids then? A dipeptide. Okay? The linkage of about 3 to 10, and we're actually going to say 3 to 20, would be called an oligopeptide. Remember, we use that for saccharides. Oligopeptide. And then what would the linkage of many amino acids together be called? What was our prefix for many? I heard it. Polypeptide? I heard someone say poly, but they didn't say it loud enough to. Okay? Generally, Functional proteins will be composed of at least 50 or so amino acids, but usually contain hundreds or even thousands. So if it's anywhere from about 20 to 50 amino acids, we'll call it a polypeptide. But if I have more than 50 or so amino acids, we'll call it a protein. Okay? So that's our terminology. So remember again that a protein is a polymer or a chain of amino acids. The first amino acid can be any of the 20 amino acids. The second can be any of the 20 amino acids. The third can be any of the 20 amino acids. The actual sequence of amino acids is going to be called the primary structure of the protein. Okay, so you don't need to know the names of each of the amino acids, you just need to know that there are 20 of them. Okay, so I hook these together like this. Okay, and they form a chain. Now that doesn't look terribly complex, right? That doesn't look like it's going to do much. It doesn't look like I can use that to build a chemical machine. Okay? But literally inside every cell of our body, we have thousands of hugely complex chemical machines that are made out of these proteins. Right now, it's just a chain. And there's amino acid 4, amino acid 15, amino acid 18, amino acid 1, amino acid 4. They all have names, but I didn't want to put the names in there. I'm just saying that this might be the sequence of a chain of amino acids. It's just a chain of amino acids. But let's think about something. If I have 20 different amino acids, and my first amino acid is just one amino acid, how many different, if I said, I'm going to call anything that has amino acids in it a protein, if I just have one amino acid, how many different proteins would I have? If I just had one amino acid be the protein, but I have 20 different amino acids, how many different proteins could I make with 20 different amino acids?
A lot.
20, right? Each one would be a protein, okay? If I had 2 amino acids, and the second amino acid could be any of these amino acids, and I put 2 amino acids together, and they could be any of 20 amino acids, how many different proteins could I make?
40.
Not 40, but
20.
So wouldn't I be able to do like A, B, A, C, A, D, all the way to 20, and then I could go B, A, B, B. How about 400? Okay, I could do this 20 times, right? So if I went through the 20th letter here, and then I started with B again, and went through the 20th letter, and then I started with C again, and went through the 20th letter, it would be 20 times 20, right? I could have 400 proteins. If I only had 2 amino acids, if I only had 2 amino acids and they could each be one of 20, I would have 400 different proteins. What if I could have 3 amino acids? I could have 8,000 different proteins with 3 amino acids. What if I had 4 amino acids? I could have 160,000 different proteins with just 4 amino acids. Imagine if I had 100 amino acids in a protein. How many different proteins could I make with 100 amino acids? And that would be 20 to the 100th power. If I raise 20 to the 100th power on my calculator, it overflows. I can't have that high of a number. Okay, but you can imagine if I have 100 different amino acids in a chain, I could have 20 to the 100th power different proteins. How many proteins do you think there are in all of biology that are useful proteins? Just take a guess. How many useful proteins do we have in all of biology that we've seen in all of our biologic organisms? In fact, does anybody know about how many different proteins we have in the human body? We have a lot, but we have somewhere between 30 to 100,000 different genes, and those code for about 100,000 proteins in our body. Okay? Now, a lot of those are similar proteins to what we see in other types of animals and other plants. So, an estimate might be that there are about 1 million different proteins in all of biology. 1 million different proteins in all of biology. But we have the possibility of having 20 to the 100th power proteins in all of biology. What does that mean for most amino acid chains? It means for most, they probably don't do anything constructive, right? They don't do anything useful. So a very, very, very tiny percentage of all of the possible amino acid chains actually do something useful. Okay? Contribute to life. A tiny, 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 tiny fraction out of all of the possible amino acid chains do something useful. Okay? I'm just going to check out something here. Okay? So I was just checking to see 20 to the 10th power is 1 times 10 to the 13th proteins. So that's about 100, that's about a quintillion. So even just 10 amino acids gives us the possibility of about 1 quintillion different proteins. But only about 1 million out of those actually do anything. So just something to be aware of, how extraordinarily incredible life is, that it selects those million proteins out of these gazillions and gazillions of possibilities. Okay? Now, this is the primary structure. This doesn't do much. It's just a chain. It's not forming some kind of an incredible machine, right? It's not a three-dimensional structure. There's also a secondary purpose for the amino and the organic acid groups in the peptide bond. Okay, it's not just so I can hook these together. If I wanted to just hook these together, I could use OH groups, right? I could just do anything to hook them together with OH groups. Recognize that a protein can't do much in the shape it is in. It's just a chain of amino acids. It's only when the amino acid forms itself into a complex three-dimensional structure does it become able to perform its designated function. So how do these things create this three-dimensional structure? Nitrogen and oxygen cause the region of the peptide bond to be very polar. Okay? We talked about the importance of polarity in water molecules. We talked about the importance of polarity in cell membranes, right? Because fats are not polar, they can't mix with water, and that's what makes them a good cell membrane, right? They can separate water on one side from water on the other side. But in proteins, these amino acid regions, the regions between the amino acids right here, their linkages are very polar, and they can attract other areas of the amino acids that are also polar. And that's the second reason for these amino acids having nitrogen and oxygen I can pull, I can attract this area, or I could attract this area. I can create these forces of attraction, and so it coils the chain up into its three-dimensional structure, and it always does it in the same way for a particular protein. It coils these forces of attraction, pull it into a three-dimensional structure. And remember, these are called hydrogen bonds, and they pull the linear acid chain into amino acid chain into a nonlinear three-dimensional structure. And we're going to call this the secondary structure. This is the second level of structure. The first level of structure is just the chain.
Dr. Marshall.
Yeah. Hi.
I was just wondering if you had a second if I could grab the packet that I used yesterday.
In which class, remind me?
B6 chemistry.
B6 chemistry. Ah, yeah. I can go to work with this.
Oh, and the paper. It was a packet.
Yeah. Step. You'd think it would be near the top of something. Here's the other assignment. So that's a review assignment, and that's the homework.
And then the whole thing.
Yeah, so I said Friday, but I'll give you an extra day. So you can have it due Tuesday.
Okay, thank you.
Okay. Sorry about that. Okay, so anyway, these forces pull this together, and it's all because of these particular bonds, the COOH bond. So it's because of the nitrogen with the hydrogen and the carbon with the double bonded oxygen that causes this, right? This is the area of the bond that's going to attract others, things that are just like it, okay? And it pulls it into the three-dimensional shape that it's in, and that's what all of the other macromolecules don't have. They don't have this area of polarity here that causes this. So if the sequence of amino acids itself is called the primary structure, this second level of structure is going to be called the secondary structure, right? Okay, the secondary structure. So because of the way in which the forces of attraction develop in the protein, two different types of secondary structures are seen. In one instance, forces cause the amino acid chain to form a special type of helix. Okay? We're going to create a special type of helix, and this is just showing all of the atoms together creating this helix, and then this is adding in the forces that pull it into that helix, and this is called an alpha helix. Okay? An alpha helix. So that's one type of secondary structure. And these bonds between the amino acids hold this helix together. Okay, so where's the chatter coming from that's distracting me? A second type of structure is called the beta pleated sheet. The beta pleated sheet. And if you fold a piece of paper up into a fan shape, right, if you do this. Right, this is pleated, right? Okay, and so that's the other structure. And again, these are just atoms that are forming these different structures. Okay, so we've got a helix and a pleated sheet. So these are two different types of secondary structures. Okay, so now we know that the acid and the amino groups forming the peptide bond are responsible for the primary and secondary structures of the proteins. Now, what is the significance of the R group? That's that third part of the structure. The R group can be formed of chains or rings of carbon and other atoms, and it can have properties of weak acids or weak bases, and it can be polar or nonpolar. The significance of this is that the R groups of some amino acids can also develop attractive forces for each other. Some of these are hydrogen bonds, but there are also other weak forces that we aren't going to actually discuss. And these can pull the secondary structures into even more complex three-dimensional structures. And this is what that looks like. Okay? So I might have a linear chain of amino acids. So this is just going to be the amino acids in a chain here. But then I've got an alpha helix here because I've got forces of attraction that are pulling the chain into that. And then I've also got these pleated sheets here. Okay? And so what the R groups do is they take linear parts of this and they take helical parts of this and they take alpha sheets parts of this and they pull them into even more complex structures. Okay? That's a pretty complex structure, isn't it?
Yeah.
Okay? This is a machine. This does something to chemicals that provides a function. Okay? How many of you remember anything about the electron transport chain? Or do you just remember that it's part of cellular respiration? So you remember the electron transport chain is part of cellular respiration. What's the purpose of the electron transport chain. What do we make because of that? Yeah.
ATP.
Yeah, we make ATP, right? The last step in this is a protein called ATP synthase. And ATP synthase sits in the cell membrane, and hydrogen ions flow through it like a paddle wheel. And this protein is a literal three-dimensional paddle wheel. There's a paddle wheel inside of there that goes around and around and around. And as the hydrogen flows through it, it causes this paddle wheel to turn around and around and around, and that's what makes the ATP. Okay, so our proteins, our enzymes in our bodies are literally machines that accomplish functions, and it's only because of the amino acid, the peptide bond, and then because of the R groups that are present that pull this into even a more complex structure that we can actually make these machines that physically do things. Okay? They have arms that come up and swing out like this, or move things around, or spin around like this. There's all kinds of machine components inside of our cells that do these amazing things. And I'll show you an example of that when we talk about cellular respiration. So if the sequence of amino acids is the primary structure, and the first creation of three-dimensional structures is called the secondary structure, and that's caused by the hydrogen bonding, then if I have forces of attraction with the R groups, and I create this even more complex structure, hopefully it would make sense that we're going to call that the tertiary structure. Tertiary means third level, right? Okay? And then finally, in some proteins, we're just going to take different proteins and hook them together in a special sequence so that they can do something like an assembly line would. So in some proteins, large complexes of two or more different chains can be formed, and this fourth level of structure is called quaternary. Sorry about that. This is the quaternary structure. Not quaternary, but quaternary. Quaternary structure. Quaternary structure. Okay? So what I need you to know about all of this is summarized on this page. Okay, we went into a little bit more detail than you need. On any test that I test you about protein structure on, you'll see this diagram. Here's the primary structure, it's just the chain, right? Here's the secondary structure, it's either an alpha helix or a pleated sheet. Here's the tertiary structure. I chain these sheets and helices together in a complex three-dimensional way. Here's the quaternary structure. I put two different proteins, I hook them together in a specific way. So primary, secondary, tertiary, quaternary. All I will ever do on a test related to the structure here is I will mix these up and mix the names up and ask you to match them. Okay, so that's all that I'll do in relation to that particular type of thing. Okay, but I want you to understand how amazingly complex these things are, and there's there are entire websites that are dedicated to just one protein or another protein that you can go to and look at the actual three-dimensional structure of these things. And later on in the course, we'll actually do a little bit of that, because we want to know what some of the structures are that give rise to certain types of functions. Okay, so we alluded to the specific functions before. So the first thing on our list was structural elements, right? We have structural proteins. And the way that I remember that when I'm thinking about how am I going to remember all these different functions, when I was a kid, my mom and dad said you better drink your milk because that will build your proteins, right? Your milk will build the proteins that you need. And that always implied to me that I had lots of structural things inside my body. Proteins that provide structural support usually take the form of rope-like fibers. So if I take the form of rope-like fibers, hopefully it might make sense that I'm going to be called a fibrous protein. Okay, so how does structure represent function? How do we make a rope stronger?
We add more rope.
Okay, but how do we hook it together? How do we structure it? Do we kind of coil the rope strands around each other? We twine them together, right? Doesn't this kind of look like that? This is how our structural proteins are made. Keratin molecule, this is a tough structural protein present in skin, hair, skin cells, hair, and finger and toenails. Another example is collagen, the fibrous protein that provides the support for most of our connective tissue. So we have a lot of alpha helices in fibrous proteins or structural elements. And then they twine around each other. And then they twine around each other as well. And then they twine around each other in what we're going to call a filament. Okay? Does anybody remember anything about the cytoskeleton? Remember we have, in a cell, we have proteins that crisscross the cell that hold the cell structure together. Does anybody know what rebar is? What's rebar? Reinforced concrete, right? It's reinforced with these rods that go through the middle of them. These structural proteins are a lot like that. They are the rebar of the cell. They hold the shape of the cell together like that. We'll talk about that when we talk about the cytoskeleton. Okay? And that's what these proteins do. That's what keratin does, and that's what makes it a protective protein. It makes the cell very tough and so that it can't be damaged as easily. Okay? So structural elements, structural proteins. Now, you might say that structure is a function, but when we say functional, we actually think about things that do something physically. It causes some kind of a thing to happen, rather than it just sits there and holds things together. So we separate structural proteins from all of our other proteins. We call all of the other proteins functional proteins. So proteins that don't make up structural elements perform some type of specific function, and therefore they're called functional proteins. So fibrous proteins are usually linear and rope-like and extremely stable, but functional proteins have a more complex three-dimensional shape that is roughly spherical. So somebody got the bright idea that said, this looks like a glob. Okay, this protein looks like a glob, and so these proteins are referred to as globular proteins. Okay, and the three-dimensional structure is what gives rise to a complex function. Usually, there's part of the protein that has a special shape, and we'll finish here in just a few minutes. There's usually a part of the protein that has a special shape that only fits with certain other chemical compounds. Okay? and this part of the protein is called the active site. So I've got an active site here. And remember, we said that one of the main functions was these dehydration reactions. So if I was looking at one of these dehydration reactions, I might fit an OH group in right here, and I might fit the other hydrogen in right here, and then the enzyme will do something to cut out that water molecule out of those two and leave the other part of that molecule joined together. Okay, so the other part of those two molecules is going to be joined together now after it cut the water molecule out. So that would be an example of that type of a functional protein. That's an enzyme. So the chemical substances that fit into the active site are called substrates. Now, this is a problem. These proteins are problems because they depend on fairly weak forces of attraction to hold them together. There are thousands of different forces inside of here holding this together in exactly the right way, but these forces of attraction are easily destroyed if I have a bad pH or if I have too low or too high of a level of concentration of like calcium or sodium or something like that. So if pH or concentration of certain ions is out of balance, these proteins will unravel. Okay, come on. They'll unravel, and they will become functionless because they unravel. Also, temperature is a big thing too. If you go above a certain temperature, you're going to have problems. So if I have too high of a fever for too long of a time, my proteins will start to unravel and become functionless. I'll die. Come on. So, that's as much as I want to say about those particular molecules at this point. We need to add these things into our list. And we have enough time to do that. So, we also need to add in nucleic acids. So I would write this down on a separate sheet of paper or some kind of a blank sheet of space that you might have. We've already done our carbohydrates. And our lipids. Nucleic acids, I've told you what I want as far as functions go. And I've said we want heredity. And I've said that's DNA. And we said protein code. And we said DNA stores, RNA transmits, and now we have proteins. And our first function is structural. And the two examples I've used are keratin and collagen. Okay, so we remember 2 points, 2 points, 2 points, 2 points. for each of the main groups, 1 point for each of the functions, and then each of these that you can name is a quarter point extra credit, right? So you can get a lot of extra credit in the structural, or in the protein section. The second one we said was biological catalyst. And we said enzymes are what we wanted for that. Okay. Let me get back to that wasn't what I wanted to do. Let's stop trying that. Let me go back to the. I just want to make sure I said what. So yeah, enzymes was all the things that we had there. And then for transport proteins, and we're just going to say transport in blood. We said ferritin, and we also said transfer. I'm sorry, transfer, not ferritin. So that'll be transferrin. Why is that small now? Okay, let's try this again. So transferrin, and then transport proteins in cell membrane. That would be GLUT proteins. Okay. And I don't care what order you put these in. I usually remember them differently every time I do this. So transport and cell membrane. Cell communication. Cell communication is my fifth function there. We had glycoproteins. As that, We had immune proteins. And these were antibodies. And then we had two more. We had, so that's transport. Cell membrane receptors. Hang on just a minute. Cell membrane receptors and forgot what we said for those. Is it insulin receptor? Okay, would be an example. Insulin receptor. So insulin receptor, and then finally chemical messengers. Chemical messengers and insulin would be a good example of that. Okay, so this is what I want for your list. You know what the other two are. You know what these are now. And I'll give you 2 class periods to actually, 2 class periods, and then we'll have a question of the day on that. That would be, today's Tuesday, Thursday, so that would be Monday. So Monday, a question of the day would be the entire list. Okay?
Yes.
Okay.
Thank you.
The project we got.
Yes, you were here yesterday.
It's so stressful when you know somebody knows that you're there. Thank you, Dr. Mark.
Yeah.
Yeah, you gotta take a second.
See ya. Take care. I just had.
Alright, let's go. I taught them how to play.
Thank you, Dr. Marshall.
See ya. Peace. You both need them?
No,
I'm just here for moral support.
thank you.
So that's a review assignment.
This
for,
one?
yeah, and then that's what we're working on right now.
Okay.
Okay, if you need any help, come and see.
Thank you.